Hello, everyone. I'd like to welcome you to today's webinar from PacBio titled No Organism Too Small, Build High-Quality Genome Assemblies of Small Organisms with HiFi Sequencing. My name is Michelle Vieira, and I'm the Manager of Plant and Animal Sciences at PacBio, and I'll be your host today. So before we begin, I'd like to go over a couple of housekeeping things. So first, all attendees are muted. So if you have questions or comments you'd like us to answer, please submit them at any time within the GoToWebinar question panel as shown here on the right. We've saved some time at the end to take questions, and we'll try to answer all of them, but if we can't get to everyone, we will follow up with you after the webinar. In addition, today's webinar will be recorded and made available to you, so look out for that email after the fact. And then lastly, we always want to hear your feedback on our webinars and how we can improve. So along those lines, please consider sticking around for a couple minutes at the end to fill out a survey that will pop up as soon as we've finished. So today we have a wonderful lineup of speakers, including Marcela Uliano da Silva of the Wellcome Trust Sanger Institute, who will tell us about the low input assemblies she's worked with as part of the Darwin Tree of Life project. We also have Scott Geib of the USDA ARS, who will share some of his low input work on the agricultural pests. And finally, we have Chris Lahmer of the Wellcome Trust Sanger Institute and EMBL EBI, who will share his really exciting work on generating high quality genomes from picogram amounts of input DNA. But before we begin, I wanted to spend just a minute or two making sure we're all up to speed on what PacBio sequencing is about. So PacBio sequencing systems generate highly accurate long reads, or HiFi reads. There's often confusion about accuracy of sequencing technologies around things like read accuracy, single molecule versus multi-molecule consensus accuracy, polishing, etc. So I'll start by saying HiFi reads are long and accurate individual sequence reads. They're generated by sequencing around a DNA template multiple times, as shown here. Our SmartBell libraries create circular templates for a polymerase to sequence around. And as the polymerase goes around the molecule multiple times, it creates really long reads, repeating the original template sequence. We can then feed these really long reads into the CCS algorithm, which piles up all the individual insert sequences and calls consensus at each base. And because PacBio sequencing has virtually random errors, the consensus calling at each base washes out these errors, leaving us with a HiFi read that is more than 99% accurate to then use for whatever genomic analysis you're aiming to do, whether it be genome assembly, variant calling, or something else. So you can imagine that HiFi reads could be quite valuable to genomic analyses. But why is that? Well, first, they're long. Each HiFi read is tens of kilobases in length, allowing you to span repeats in the genome. These reads are also accurate, with each and every read being at least Q20 or 99% accurate. By nature, HiFi reads have single molecule resolution to sequence native DNA or cDNA molecules. And they're the least biased on the market today, with little to no GC content or sequence complexity bias. So naturally, they can be used for some really great genome work like the projects you're going to hear about today. However, long-read sequencing has often been limited to organisms that can provide large amounts of high molecular weight DNA. Until now, that is. With the majority of organisms on the tree of life being very, very tiny, we've worked really hard to develop methods to reduce DNA input amounts to make high-quality genome assemblies accessible for all organisms. Where the standard SMART sequencing protocols generally require micrograms worth of DNA, we have the low DNA input protocol, which reduces that requirement down to just a few hundred nanograms. This protocol is similar to the standard prep with a few changes, including no post-library size selection, that allows you to sequence and assemble genomes up to around 1 gigabase in size. In addition, you can expand beyond that genome size if you have more than the 300 nanograms of required input, but still under the standard requirement amount. Both Marcela and Scott will be sharing some assemblies generated from this workflow. We're also now in late-stage development of an amplification-based ultra-low DNA input workflow that Scott will share his beta testing experience with. This requires only 5 nanograms of input DNA to sequence and assemble genomes up to 500 megabases in size. The main thing to recognize with this protocol is that we are now in the realm of amplification, which automatically comes with the limitations of PCR, which you'll hear about from Scott as well as from Chris, who has developed a similar approach that reduces inputs even lower. So depending on your organism and genome size, there's likely a workflow that will allow you to get HiFi reads for your project. So with all of that being said, let's get to the main event, and I will go ahead and turn it over to Marcella.
Good afternoon, good morning, good night, everyone. I am Marcella. I am a senior bioinformatician at the Sanger Institute. I work in the Darwin Tree of Life project. In the Darwin Tree of Life project, we went to sequence the genomes of all of the eukaryotic organisms in Britain and Ireland, so over 60,000 species. In this project, we have a huge challenge as we want to produce the best genomes possible at the same time that we test the latest sequencing technologies and algorithms to then be able to produce this very high quality genomes and transform the way that we do biology and we study evolution, conservation, and biotechnology. So in the Darwin Tree of Life project, we are using different types of data that we can characterize here as long reads. In the example here today as, for example, HiFi PacBio reads, and also data that is not necessarily long read, but is long range. For example, like chromatin conformation captured from HiC reads or linked reads from Chromium 10X. The small things that I will be talking about today are Lepidoptera. So I will be presenting some genomes from Lepidoptera that occur in Britain and Ireland. Lepidoptera are moths and butterflies. They are one of the major superradiations of insects. They represent more than 300 million of years of evolution. The assemblies that I will be presenting today, they vary in the genome sizes. They range from 249 million bases to 391 million bases. The heterozygosity also varies from 0.7 to 2.2%. And the PacBio coverage that we have goes from 34 to 77 times the estimated genome size for this species. So all of the results I will be presenting for you today, they were assembled with this pipeline here. So we have used three types of sequencing data, PacBio HiFi reads. We have used Chromium 10X linked Illumina reads for polishing, and we have used chromatin conformation capture HiC data from Arima or Qiajian for the final scaffolding of these genomes. So because the quality of the HiFi reads are very high, even with low heterozygosity, if you are assembling a diploid genome, you are able to assemble separately the both haplotypes. So we have been using the software HiCanoe, which does exactly that. At the end of the HiCanoe assembly, you have double of the expected genome size of the haplotype of a diploid species. Then we use an algorithm called purge dupes to separate these haplotypes in a primary and haploid assembly. It's not perfect, but it's one of the best methods so far if you don't have a trio assembly in your hands. Then we are concatenating both primary and haplotigs here and running one round of polishing. So we align reads with long ranger, the linked reads from Chromium 10X, and we call consensus with freebase. And finally, we take only the primary contigs here and we scaffold finally to chromosome level with the Salsa software and the Arima or Qiajian HiC data. So all of the 4 genomes I'll be presenting to you today, they were assembled with this pipeline. Okay, so let's go for the first genome I want to show you. So this is Vanessa Atalanta, the red admiral butterfly. Let me walk you through the slide here. So here in gray, you see the composition of this genome. So it's in K-mer analysis of the composition of the genome that was plotted with the high quality reads. So this is a straightforward advantage of the PacBio HiFi is that you can study the composition of your genome straight away from your PacBio HiFi. You don't need to wait for any Illumina data to be able to understand the characteristics of your genome. So Vanessa Atalanta here was estimated to be 359 million base pairs. The heterozygosity is 1.1%. The repeat fraction is not very high. So according to the K-mer is only 10% of the genome. For this assembly here, we had 34 times coverage of the PacBio HiFi, and all of the data produced for Vanessa Atalanta in PacBio HiFi reads had 11,000 base pairs or longer. So that's the N50 of our long reads. And then here for our results, so we have assembled it to very close to the estimated genome size. we have assembled it in 212 scaffolds, where half of the genome of Vanessa Atalanta is in scaffolds that are 12.58 million base pairs or longer, which is our scaffold N50 here. So one thing that is also of notice here is that our numbers of contigs, which are the sequences of DNA that don't have any ends in the middle of them, it's very close to the sequence, the number of scaffolds that we have for this species here. So if we have a look at this HiFi heat map here, in the diagonal, we see each of these 212 scaffolds count here. So as soon as we see this like very red dense square here is one of the scaffolds, and the density is the high C reads mapped back to the scaffold. So things that you see off of the diagonal can be different things. For example, if sex chromosomes that have homology between them would share high C reads, then you would see reads mapping to two different scaffolds here. And sometimes also you see errors on scaffolding. As you can see from this heat map here, the heat map's pretty clean. So most of the scaffolds here is assembled to chromosome size, or it's assembled as an entity, so it's well assembled. And we have a few things here to be manually curated. Another advantage of the PacBio HiFi is that you can pop out the mitogenome from your assembly. So here we have the mitochondria of Vanessa Atalanta that was assembled by the HiCanoe. Because of the circular nature of the molecule, it was repeated many times. So I had a small pipeline just to take out the redundancy and do a quick annotation of the mitochondria to check if everything is okay, but the mitochondria is there. Last but not least, we can also talk about gene content. So not only structure of the genome is very good, but if we look at this Busco insecta, so basically it's expected that every insect assembly is going to have this amount of orthologs presented on it. It's not 100% like this for every species, but it's a good measure of gene composition. And as we can see, Vanessa Atalanta has 99% of the 1,658 orthologs are supposed to be in insecta, so that's pretty good. I want to show you here more about Vanessa Atalanta. So as I told you, we are able to assemble both haplotypes of this genome, and here you see the numbers for the primary and for the haplotigs. You see the total length is very close. So we have the two haplotypes of this assembly that you can also notice here in this Lahmer plot. So here, basically, we have plotted K-mer analysis between the K-mers presented in each of the primary and the haplotigs, and the K-mers that are present in the Illumina Chromium 10X reads, which are also very high quality. So as you can see here in green, we have the homozygous K-mers, which are the K-mers that are shared by both haplotypes in this diploid genome. And here with half of the coverage, we have the haplotypes for each of these assemblies here. In gray, you see K-mers. There are errors, which are not very frequent, and they are not present in our assembly. So that's exactly what we want. So you can see a nice plot of our K-mers here for both assemblies, and the QV is really high. So PacBio HiFi assemblies are giving us very high quality per base for these assemblies. Finally, the amazing team from Kerstenshove at the Sanger is looking through our genomes and curating. Generally, they have been finding that these genomes are very well assembled, and as you can see, they have resolved the issues that still remained. So they have placed Vanessa Atalanta in 33 chromosomes, and 99.44% of the bases are in these 33 chromosomes. So the scaffolds remaining here represent less than 1% of the bases of this genome. So it's pretty well assembled. So I will show you a few more then. So it's similar slide. So here you have the composition of the European peacock, heterozygosity 0.7. It was assembled to close to the estimated genome size. Our N50 is 12.94 million base pairs. As you can see, again, the heat map of the HiFi is pretty clean. Only a few things here to be resolved. the mitogenomes there, our Busco insecta is really high. And then we have the same here. We can see primary and haplotigs. The lengths are, so both versions are pretty complete. The QV is really high, 60 here, and the curation team has resolved the final issues with this assembly, and we have more than 99.9% of the bases in the 31 chromosomes, which is the expected karyotype for this species. Finally, we have the aeroprominent moth. Similar thing here, it was assembled to the expected genome size in 206 scaffolds. Our N50 is 11 million base pairs, so half of the genome is in scaffolds. There are 11 million base pairs or longer. Heterozygosity is a little higher here, 1.5. As you can see from the heat map, it's also pretty well assembled, pretty clean, has a good Busco score. The mitogenome is also present here. Same thing, we have a good separation of the primary and the haplotigs, as you can see from the numbers and from the K-mers. Here is just to talk a little bit about the QV. So even before one round of polishing, the QV for these genomes are really high. It increases slightly after one round of polishing, and then that's why we have been using it so far, but further investigation is needed to understand exactly what's going on with the polishing, what is being changed. So here, as you can see, also curation team put the last pieces together, and we have more than 99% of the bases in the 31 chromosomes of the species, which represents the karyotype for the species. Finally, we have the small white. Similarly, we have assembled to the estimated genome size. We have assembled it in 45 scaffolds with an N50 of 10.6 megabases. And as you can see here, the heat map's pretty much done. The mitogenome was also pulled out of this assembly. We have a good separation of both haplotypes, and we have 99.84% of the bases in the 26 chromosomes, which is the karyotype for this species. And here, just to show you, so we do see an increase in QV after one round of polishing, which is the final QV being 58. Right. So some final considerations here. So as you could see in these 4 assemblies that I have shown you today, the HiFi reads have a very high quality, which allow us to plot K-mer analysis to understand the genome composition of the genome straight away. This can seem very trivial, but for a bioinformatician, this is really important, especially if you are trying to study a species that you don't know very well and you don't understand how large the genome size is, what is the heterozygosity. So if you do need to wait for another type of data, apart from your PacBio data, to understand it, it can, you know, it can make your work harder. So it's pretty good to be able to plot this K-mer analysis from there. So our Lepidopterans assembly were assembled to the chromosome level, which is really exciting. So we have all the species with more than 99% of the bases into chromosomes. We have evidences for telomere to telomere. I haven't shown you that, but we also have found the sex chromosomes for this Lepidopterans. About the polishing discussion, so what I have been seeing is that after one round of polishing, the genome size decreases a little bit. So it seems to me that what's still present in the PacBio HiFi reads are insertions. So even before polishing, the quality is really high. And I'll be investigating further exactly what's being changed with the polishing. We have results from collaborations from Sergei Koryan, for example, that he has a study in the human genome against Bach sequences that represented the true sequence of certain repeats that some polishing can sometimes, I mean, polishing with PacBio actually can sometimes mess up dinucleotide repeats in these regions of the human genome. So this is just to say that we haven't been applying polishing with the PacBio itself. We have been applying polishing with Illumina, which has been increasing QV and sometimes Busco scores. And exactly what's going on, what remains that is being changed by this polishing is going to be investigated further soon. I have presented you 4 assemblies today, but we have many more. So please visit us in this GitHub, which has our interim data. All of the genomes that are curated will be present in ENA and in the NCBI, also can be downloaded from there, but we are more than welcome to come to this GitHub and download all of our data and assemblies. Please use it. We'll be glad if you do so. I need to thank everybody that works in this project. So it's so many people collecting samples, sequencing them. I'm only here presenting today because we are a very big team, and that's why I'm being able to study such nice sequence data. Especially I want to thank my manager, Shane McCarthy, with whom I talk about genome assembly on a daily basis. If you want to talk to me, you see my email here on the top, my Twitter, please write to me. I thank you all for your attention. Thank you very much.
So yeah, so I'm part of the Ag 100 Pest Initiative. It's a large group, consortium with many steps. And we're selecting from the top 400 nominations of across at least 10 different insect orders. These nominations are coming from places like USDA APHIS, our regulatory agencies, ITAP and CAP surveys, USDA researchers, and then the Insect 5000 Genomes community. What's different about our project is it's a pretty diverse nomination, including pests of field crops, animals, bees, forests, stored products. Some of these are pests of foreign pests that aren't necessarily here yet, but are considered to be highly invasive to the US. And so really it's a community thing. And if you do have, if you work on insect pests and have interest in getting their genome sequenced, please reach out to us about that. So one of our real goals is how do you sequence every pest insect? Ideally, we'd like to target chromosome scale diploid assemblies for these pests. This requires multiple library types, like talking about in the last talk. We're focusing now on getting the contig assembly solely from PacBio HiFi data, and then scaffolding information from things like HiC, BioNano, or linkage maps, depending on the critter and what's available for it. We'd like to target chromosome scale diploid assemblies for these species, following the EBP quality standards, which is 3.4.2, meaning a 1 megabase contig N50, 10 megabase scaffold N50, and then chromosomal assignment, and reaching the QV40, which we think is pretty achievable easily using just the HiFi data alone. Some of the limitations for us, though, are small physical size with limited DNA material for various methods, and extremely wide diversity of taxa, types of tissues, size, everything. So we're sequencing from big happy beetles where you can get micrograms of DNA to things that you can't even see are present in your tube. So a large proportion of our samples fall below the typical standard library prep amounts. There's been massive changes in the last several years towards improving low input samples in relation to PacBio technology. Things like moving from the Sequel 1 to the Sequel 2, the 1M to 8M cells. So this is with only modest increase in loading mass, so the actual molar ratio or molar mass you need to load. You can get a huge increase in read yield. And so this is really important for samples with low input. So going in with just a little bit of library, we can get many gigs of data. We were transitioning from CLR long reads to these HiFi CCS reads that were talked about earlier. CLR targeted the longest reads possible. So when you're targeting these long reads for the same nanogram amounts of DNA, you're actually having a lower molarity of your sample. So by HiFi targeting shorter fragments, you actually get a higher molar concentration of a sample in the same nanogram amount. So for example, a 15 kb library will have 3 times lower or higher molar concentration than a 45 kb CLR library. And then some improvements we made in low input library preparation, things like precise shearing of samples to target the subread lengths that we want, removing the gel-based size selection steps that have high sample loss, things like Sage's Blue Pippin. Size selection does a very good job of creating the size fractions you want, but you usually get greater than 50% sample loss. So With HiFi reads, you are getting shorter subreads with lower nanogram amounts of library. So now we can target smaller insects. We're getting highly accurate CCS reads, which are required at a lower coverage to still create a highly contiguous assembly. And additionally, the assembly of these HiFi reads is much faster, which is really important for large sequencing initiatives where you don't want each assembly to sit in queue for say, a week, chugging through the process. So some of the things we need are methods for accurate shearing of low concentrations of DNA with little off targets, which yields the amount of library that's doesn't allow for sizing. So just to give you an idea of that, this is a low input HiFi subread library we prepared. You can see the input genomic DNA was very good. Very little tailing on the side here, so most of it is greater than 45 kb. Shearing on the megaruptor, you can see we got a very nice peak here at 17 kb, and more importantly, very little shouldering on the side. So the final HiFi library made from that replicates this size range very well. And then we do have there's an ampere step in the process which removes fragments shorter than 3 kb. So while this isn't maybe as tight as what you would get from using a blue pippin run, this library is sufficient for creating nice long HiFi reads without size selection required. So with no size selection, you have no sample loss. So we're able to start these with hundreds of nanograms of DNA and go into the sequencing with hundreds of nanograms of DNA. This is a typical run. This is HiFi data, so you're getting hundreds of gigabases of total DNA. But when you look at unique molecular coverage, you're getting 30 to 50 gigabases, which would represent over 100x coverage of something that's 300 megabase in size. So tons of data still. And you can see our means and N50 subread links match very closely with the sizes that we were getting based off of the shears. So what kind of assemblies do we get out of this? Very good assemblies is the answer. This example is Indian meal moth. Here, this was one of our earlier HiFis where we weren't quite focused on targeting the longest reads as possible. So our subread was only 12.8 kb, generating 52 gigabases of unique coverage or subread coverage. This organism is thought to have 30 autosomes plus sex chromosomes, and assembling it with HiFi ASM gave us a very, very high quality genome. So there's 14 pieces of the genome with an N50 of 8.9 megabases. But I think more interesting is there's only 35 contigs with an N50 of 3.4 megabase containing 90% of the assembly. And so if it only has 31 chromosomes, you basically have this in 35 pieces at a multiple megabase scale. So if we validate that assembly, no slides, so I gotta scroll down here. Using some HiC data, this is from Arima Genomics, you can see very strong support for the HiC data, and then each of these little green blocks inside is a contigs that are spaced inside of the HiC. And so you can see there are many chromosomes that are supported by a single contig going end to end on it. So very good results from this, very nice place to start when working with the HiFi data. So you're really looking at assemblies where you're talking there's 61 contigs that are greater than 50 kb, resulting in 99% of 0.74% of the genome. So you're not starting with thousands and thousands of contigs that you're placing anymore into chromosomes. And just seeing it a little bit clearer there, you can see the chromosomal, the contig layout onto the chromosomes. So then we move to these weed feeding beetles that are released for biocontrol doing 4 species. Each of these has about a 425 megabase genome size. Again, we got these very high quality assemblies, either with HiFi ASM or HiCanoe, with tens of megabase contig N50s. and a very large proportion of the genome contained in fractions 50 kb or larger. So those are still considered low input because we're not getting sufficient amounts to run size selection on a SAGE system with. But we have sort of truly low inputs, things that yield low amounts of DNA, but also have low genome sizes. So here we wanted to try, this was before we were doing some of the ultra low amplification based technologies, but things like copra beetle, tribolium, the cigarette beetle, they're only millimeters in length. You're only getting a few hundred nanograms, if that, for library prep. But we were still able to get good sizing, good shearing, but just didn't have the molar ratios to do to load a cell. So to resolve that, we just barcoded multiple libraries onto a single cell, and that allows the molar loading concentration to be met. And again, these assemblies continue to be very strong. For example, with the tribolium beetle, with an N50 of 14 megabases. So in addition to having many samples that are below the standard input levels, needing micrograms going into a standard approach, and so we're using the low input non-size selection approach. We also have many pests that are very tiny, so much below this 100 nanogram DNA yield. There's a lot of beetles out there that are tiny, thrips, whitefly, mites, mealybugs. You can keep naming insects, and they're small and don't yield a lot of DNA. So we tried beta testing this amplification-based ultra-low technology, and really think it's going to be critical for the success in these species and in our project as a whole, really if we want to maintain these single insect diploid assemblies. So going into examples of even tinier beetles, talking here about the sawtooth grain beetle, just had another one finished an assembly last night as well within the same genus. These are not super tiny, they're about the size of these cigarette beetles or this copra beetle, but they're very, very flat. And so they just don't yield a lot of DNA. We're probably talking between 60 to 80 nanograms of DNA. So we tried the standard input approach and pooling it with others, and we just weren't able to get enough coverage to achieve a good assembly. So our N50 was low, below 100 kb, and our BOOSTCO kind of left something to be desired, 92% complete with 6% missing. Doing an ultra low library prep, starting with 5 nanograms of starting material. So then we take that material, ligate on the adapters, amplify it, and then have sufficient material for then shearing the library and or making the library and then size selecting it for the fragments we want. We got a much better assembly with an N50 of 1.87 megabases, and our longest contig now is over 7 megabases in size. And more importantly, the BOOSTCO scores here are much stronger. So even though we're using amplification, which may miss regions of the genome because of difficult to amplify regions, we're seeing essentially a complete gene set here, a very strong contig assemblies. And so we really think this is a good option. And just to talk about the one that was completed last night, the N50 actually eclipsed 2 megabase. I think it was 2.8 megabase N50, again with a very high BOOSTCO scores. So we've made libraries for other insects, things like thrips and whiteflies and mealybugs, and they're in queue for sequencing now, and we'll be happy to share those results when present. So in conclusion, essentially any insect, if you can get more than 200 nanograms of DNA, high quality HiFi data can be generated from it. Using these low input approaches, new single insect assemblies can be made mostly routinely, allowing true diploid assembly, so assembly of both haplotypes. Even in insects with higher yields, you may partition tissues for different techniques. So if you have a fly or something like that, you may just choose to take the thorax, make the HiFi PacBio data from, and save the rest for doing things like HiC or something to make chromosome scale hybrid assemblies from a single individual. Shearing, again, has become very important to generate tight reproducible shear profiles. to allow for this high quality sequencing when you're not size selecting. And then very tiny samples are becoming possible with ultra low methods. So we're going in with 50 to 20 nanograms input. Potentially there's some reduction in assembly contiguity. The examples I showed, the N50s were in the multiple megabase rather than tens of megabase from the related beetles in the slide before. But we're still seeing very high BUSCO scores. So the gene spaces there, it's probably breaking down in AT or GC rich regions that are repetitive. Yet to be tested would be the very tiny insect with a very large genome size, but ultra low library approaches provide ample library to generate sufficient data for this. So we can start getting into that and looking at the impact of amplification on contiguity. So even from this ultra low approach, you're able to amplify to nano or microgram amounts of DNA that should be able to be run on subsequent cells to generate higher coverage if needed. So with that, I'll just thank the sequencing folks within ARS or in Clay Center, Nebraska and Stoneville, Mississippi. The insects I talked about in stored pests and weed biocontrol today are championed by these folks. And then we're doing extractions and library preps in Hilo, Hawaii with the team there. And we have a large assembly group that works to putting these great assemblies together. Thanks.
Good. So I'm Chris Lahmer. I'm a postdoc at the EBI and the Sanger Institutes here in the UK. And I'm very grateful to PacBio for inviting me to talk about my wet lab method development work, which is focused on understanding genomes in the world's smallest animals, which are the myofauna. And that's a word that maybe not everyone's heard. So it's a very practical term. It means organisms that are smaller than a millimeter, that'll pass through a millimeter wide sieve. There are lots of organisms like that, many parasites, many soil fauna. But for me, the canonical myofaunal group sort of environment is the marine interstitial habitat. So the spaces between grains of sand in the ocean. And the organisms that occupy this habitat are really heterogeneous taxonomically. They include more than 20 of the so-called animal phyla, and there are some major animal groups that only occur in this kind of habitat. But they're very coherent ecologically. That is to say, they're really well adapted to living in these tiny spaces. And it's a complete ecosystem into itself. There can be many kind of grazers, mostly on biofilms, scavengers, predators, chemoautotrophs, organisms with photosynthetic algae, and the organisms that live in this habitat mostly complete their entire life cycle within it, meaning that they have pretty poor dispersal abilities, and they also have really short life cycles. And you can see that many of them are strongly adherent, maybe have long tails with sticky glands on them. Many of them are quite elongate. And this poor dispersal ability and their short life cycles is probably one of the reasons that these are extremely speciose. So diverse not only at the phylogenetic level, but at the species level. Outside of Europe, it's rare to sample described species in the myofauna. And when you start looking at molecular taxonomy, each morphological species very commonly has multiple clear molecular lineages within it. And when people have started applying meta-barcoding approaches, you can often see hundreds of OTUs within a single kind of habitat. And so this has led to speculation, although this is highly uncertain, that these organisms have potentially millions of species here on Earth, which means they're one of Earth's major reservoirs of biodiversity, and we should be thinking about them as much as we are coral reefs or tropical rainforests. But they're really hard to study because they're so small. One organism might have only a few hundred or a few thousand, maybe a few tens of thousands of cells, if you're lucky. And they often have quite small genomes, although that varies too. And you can do some back of the envelope math to show that a 1,000-celled organism with 100 megabase genome only has, around 200 picograms of DNA that you can possibly extract from it. and that's thousands of times less than you need even with the low input PacBio protocol. So studying the genomes of these things has been tricky, and one of my major projects as a postdoc has been developing new methods to tackle this. I'll give a brief layout of that method that I've converged on. If you want more detail, I encourage you to look at the Smart Leiden talk that I gave a few weeks ago, which should still be up online. And we're also, preparing this for publication very soon. So hopefully this will be accessible to everyone. We're making a protocol document as well. But in brief, what we do is isolate the specimen, hopefully in a relatively clean state. You might starve it overnight, for instance. Then we use a homemade salty lysis buffer, TNES, to bust it open, put all the DNA and RNA into solution. One optional thing that I don't have much time to go into, but is very important actually, is that this protocol is also compatible with getting full-length cDNA libraries from the same organism that you're doing a genome assembly from. That's through magnetic bead hybridization and template switching reverse transcription and PCR. And that's going to be really important for genome annotation, obviously. But anyway, you've lysed your specimen. We then do a salting out DNA extraction using a yeast tRNA carrier, and that really drives precipitation of the DNA efficiently. So it's a relatively quantitative extraction. What you put in, what you get out is what you put in. You then have your purified high molecular weight DNA at low concentrations. It's very fragile, so you want to process quickly, handle delicately, yada yada. One other optional branch that you might take in the protocol is to take a small aliquot of the DNA, QC it on a platform like a FemtoPulse, and then prepare a linked read library. I've used the 10x Chromium solution. Unfortunately, that's off the market now, but I think there are a number of replacements that should work for perhaps all but the smallest organisms. The important thing is to make sure that aliquot gets on the order of 300 haploid genome equivalents, similar to what you'd put into a 10x, so that there's enough diversity to generate a lot of linked reads without many PCR duplicates. But most of the DNA, 90% of it, should go to your long read library. We shear it using a megaruptor 3. That gives us a really, really tight distribution of around 15 kb, so that minimizes the loss of potential sequenceable library. And then we put it into a library prep process. I've used NEB's Ultra 2 kits for that. And that gives you kind of a familiar Y-adapter tagged double-stranded library, but you still only have a few hundred picograms of that, so you have to amplify. And I use a really carefully optimized long-range PCR, 20-minute extension cycles. Only a few polymerases do really well for this sort of thing. Another trick that's been really important is I've learned to take a very small aliquot of that finished library, amplify it in the presence of a dye, and then use sort of the inflection point of that fluorescence curve to estimate how many cycles I would need to amplify the remaining 90, 94%. And that's really helpful in not overcycling, which can ruin your experiment, or undercycling, which can be annoying and also ruin your experiments in some ways. You usually get a distribution of DNAs a bit like this. There is a shoulder, unlike what Scott was showing, and I think that's just because low concentration high molecular weight DNA is pretty fragile. And those shorter reads would contaminate your sequencing run and give you shorter reads than you'd ideally like. But you have lots of DNA at this point, hundreds of nanograms, even micrograms if you like. And so you can do some size selection and then put that on your long read sequencer of choice. You can even do your long read library prep and then size select if you wish with PacBio. Okay, so how do we know this works? Although I started with in vitro diluted DNAs, I eventually moved to real organisms, and I used the worm that we know and love, C. elegans. And here's some data from a single C. elegans worm that we collected last fall. So we got around 1.2 million HiFi reads. There was a little evidence of DNA damage from the FedEx shipment, so I don't recommend that being a standard part of your workflow. But we nonetheless got almost 14 gigabases of output and a mean read length of around 11 kb of HiFi reads. So that looks like perfectly viable for a C. elegans assembly. 2 different assemblers that we've tested gave us roughly the same kind of assembly, so a little over 350 contigs, an assembly size compatible with expectations, a little larger than the 100 megabases we expect from C. elegans, but there's actually a bacterial symbiont in this library as well, which is making that difference. But the N50 is not the sort of scale that Scott was showing, so more like half a megabase. The gene set is pretty complete. The Falcon assembly gives us 97% of the nematode busco sets. But this isn't perfect, and you might be wondering why. So the obvious culprit might be amplification bias. So here are some plots of that amplification bias in this and other libraries that have gone through amplification with this method. So we divide the genome up into equal-sized bins and then order them by their GC contents, and then look at the sort of distribution of coverage across those bins. That's what's plotted here. And you can see that actually across a pretty wide range of GC contents in the genome, the coverage is very similar. It's only at the very extremes towards 20% or 70% that you begin to tail off, but it's actually rare to see complete dropouts even in those extremes with this amplification method. One important thing is the level of amplification that you do does increase this bias, right? So that 23 cycle library has a little bit poorer distribution in the extremes, and also to contrast, a different organism here, Phenostomum flatworm that I've sequenced that only needed 14 cycles. You can see the distribution of coverage is very flat, actually, even in those extremes. And so the lesson is kind of the least number of PCR cycles that you can get away with is the targets. However, I've spent a lot of time as well looking at read pileups against the C. elegans reference genome. That's what's plotted here. And I, not super frequently, but occasionally see pileups that look like this, a trough in coverage. And I'd go to the center of that trough and look at the sequence, and it wasn't very GC biased at all, actually. It was pretty similar to an average part of the genome. And so I was scratching my head for a moment before I realized, oh, this is a secondary structure. This is an inverted repeat, a long palindrome, whatever you want to call it. It's a sequence that at the single-stranded level is capable of forming a strong hairpin structure. And the hypothesis is obvious. That hairpin structure is too tight for the polymerase to open it up and get to the other adapter sequence at the other end. And so that causes this dropout in amplification. Another thing I've occasionally seen is a case like this, where you don't actually have that much of a coverage bias, maybe a little bit, but in a minority of reads, you'll see an indel variation, a pretty large one. And this isn't at the ratio that you'd expect for a heterozygous variation. And so that suggests it's something that was generated during library amplification. And what I think is happening is a template switch event where the primer skips from one end of the hairpin to the other and causes a deletion that's propagated onwards in PCR. You can have both things happening at once, and you can imagine how that might complicate assembly. Another place where you sometimes see this kind of bias is in very simple microsatellites, so dinucleotide repeats. Here, it's a 30-base AT dinucleotide in the reference, but in my amplified library, it's a drop-off in coverage, and the few reads that do cross this region show insertions with hundreds of bases of AT, so that's another probable library amplification artifact. And we also see some of these effects around CT dinucleotide repeats, although I think this is a PacBio specific thing because I have very good coverage of the same DNA in nanopore sequencing. So that's one of the few areas where PacBio still has some work to do. Okay, so we can do this in organisms and culture. Can we do it in things from a beautiful temperate rainforest or whatever kind of habitat you might want to sample? We can. So I've spent a lot of my life squatting in swamps and rivers, looking for microturbularian flatworms as part of my PhD. And so I thought, let's try this. And there's this beautiful habitat, which is actually just a 13-minute walk from my my lab. And so I thought that's a perfect target. And one of the more common organisms in that habitat was this rhabdoceal flatworm called Castrella truncata. It's a member of the most diverse order of free-living flatworms, probably a kind of grazing organism common on aquatic plants. And you can see this is actually the organism that I sequenced that you have the picture of here. It's, just under or maybe just over a millimeter long, and probably had only a few thousand cells. And there's no a priori information about this organism's genome. So we put it through the process, got all three data modalities, long reads, cDNA, and linked reads with 10x. And one SQL 2 cell gave us around 2 million HiFi reads with an insert length of 10 KB. We're nowadays getting closer to 15 KB, and 19 gigabases of HiFi output. Putting those raw reads into Genome Scope, we realized we kind of lucked out because it's a very small homozygous genome. The estimated size was about 225 megabases, and the heterozygosity was estimated at 0.2%, which means it's just twice the level of heterozygosity that we humans have, and we're famously inbred organisms on the grand scale. So to get a final assembly, I used Fly in its metagenome mode for reasons that will become clear soon. And that gave us an assembly of around 237 megabases, 764 contigs, and an N50 of 733 KB. Some of that was probably haplotypic duplication, so I used that purge-dupes pipeline that Marcela introduced. That only dropped about 64, exactly 64 of our contigs, and didn't change the overall assembly span very much, so that's good. And then the thing that actually made a lot of difference was adding the linked read data using this SCAF10x pipeline. And that brought us from 700 contigs to 370 scaffolds and increased the contiguity of those scaffolds to 2.7 megabases, which is pretty good. Evaluating the completeness of the assembly using the Busco set, we have 85% of the metazoa set represented and a relatively low duplication rate, which might seem low at first glimpse to a vertebrate-centric person, perhaps. But actually, that's pretty typical for the flatworm genomes that are out there. Everyone that I'm aware of has a Busco completeness in the range of 80 to 90%, and there are even some chromosomal level assemblies that are sub 80% Busco completeness. So I think that's just a flatworm thing, and maybe we should be reevaluating our Busco sets. Finally, it's important to note that this assembly contains more than just Castrella truncata. So this is a blob plot. Each dot is a scaffold, and they're scaled by the size of the scaffold. And then they're ordered along the x-axis by GC content and on the y-axis by coverage, read coverage in the library, and they're colored by the closest hit in the NR database. And what you can see is that there's this main population that's clearly Castrella truncata. It's not all hitting flatworm, but that's because there really aren't many things close to rhabdoceals in the database. But you can also see there are these smaller populations of mostly pale green proteobacterium hitting scaffolds. And so I think these are kind of skins of the microbiome of Castrella, or perhaps environmental contamination that made it into my DNA extraction. And it's not appropriate to put them in the assembly, but we can cut them out. And when we do, we end with a final assembly of 251 scaffolds, 230 megabases, and a scaffold N50 of 3.1 megabases, which I think is pretty close to what you get with an unamplified library. So I'm really very excited by that result. Those with keen eyes will have also noticed this one scaffold, which is a little more GC poor than the rest of the Castrella genome, but pretty big. And that's actually, that's real. That's a putative bacterial symbiont, a closed circular genome of 2 megabases, which has, I think, 33 predicted tRNAs. And that's exactly what you'd expect for a living organism with its own biometabolism. And when we take the 16S sequence of that putative symbiont, it places it clearly within the phylum Rickettsia, known for Wolbachia, for instance, and particularly in this putative family Midichloriaceae. and that's a group of mostly intracellular symbiotic organisms, which has included species that have been found within ticks and insects and many aquatic and marine invertebrates, some protists, and now for the first time, I think, from flatworms. And so there's clearly some kind of biological interaction between these two organisms going on, and I think we'll only see more of this as we apply such methods to these tiny animals. So in conclusion, with long-range PCR library amplification and many optimizations, it is possible to get really highly contiguous assemblies from individual organisms of Earth's tiniest animals. It's really important to try to scaffold those assemblies because there are amplification-driven dropouts from DNA palindromes and microsatellites and that sort of thing. And also, I think it's going to be a general thing that when we sequence such organisms, contaminants and symbionts are going to be magnified in importance in the library. So it's really important to apply appropriate strategies to identify and partition those. The next obvious step is to get the whole hog, right? Try to go for a chromosomal level assembly. And the clearest path to that is to apply some orthogonal data sets like HiFi or genetic maps. And I'm thinking and working actively on that right now. So it's open season now for meiofauna. There's a lot of these organisms out there and probably a lot of really exciting biological stories to discover. And I hope that you know, this audience and beyond are going to drive that kind of work. And I'm really excited to see what it does. So that's my presentation.
Great. Thank you, Chris. So I'd like to invite the rest of the speakers to go ahead and join me on webcam and unmuted so that we can take some questions that have been coming in throughout. All right. Great. So we will start with a couple questions from early on, some specific questions for Marcella specifically. The first question we have is, how were you getting your genome size estimates that you were referencing? Was this from the PacBio data, or were you using something else like flow cytometry? Marcella, I think you're still muted.
So we have been estimating with the PacBio and with the Illumina. We also have a compilation of other people that have done flow cytometry, but just to confirm or to compare, basically do it from Illumina and from the PacBio HiFi.
Okay, great. And on the Vanessa Atalanta genome, it was noticed that the scaffold size was larger than the contig size for the PacBio only assembly. Can you speak to that?
That's the number, right? Or is it the N50 itself? Let me have a look. So is that the number? So the number is so it's actually 210 scaffolds and 212 contigs. I guess that's what they mean, right? Yeah.
Yep. Okay. One question, and I'd be curious to hear kind of everybody's response to this one, about the efficiency of doing these assemblies. So how much time is it taking to actually run these HiFi assemblies compared to previous efforts? So maybe we'll start with Marcella.
Compared now, it's interesting. I mean, I haven't run a butterfly CLR, but it's pretty fast. Can I give you exact numbers? So not more than 80 giga of memory, for example, and we give it, yeah, and it takes a few hours. I don't want to say how many hours, but a few hours, less than a day.
Okay. Scott, has that been your experience as well?
Yeah, and then also, you know, HiCanoe would still be running on a grid, but things like the IPA assembler or the HiFi ASM we're running on a single node, just giving it the cores available on that node, and it's finishing in hours for something with small genome sizes. The other thing is not to overwhelm it with coverage. You don't need 100x coverage, and if you give it that much, it's going to take longer. it'll still finish and be correct, but you know, downsampling your data in some of these cases can improve the assembly runtime.
Yeah, I guess it depends a lot on the assembler and the level of parallelism that you're going to apply. For rapid algorithms like WT-DBG2, you know, less than an hour is sometimes possible with lots of threads. I've also run Canu assemblies that have taken a day and a half, but that'll also depend on the cluster load. It's rare to see something that takes longer than that for at least the sub 500 megabases genome.
Yeah. Yeah, a big bottleneck is the correction of reads, right? Erroneous long reads. If you do need to do the mapping all versus all to correct, then it would take a long time. But as these ones don't have that step, then it's much faster.
Yeah, although the CCS algorithm isn't instant either, though.
Yeah, so there is that. Yeah, it's true.
Okay, and then this question I think originally was for Marcella, but I think it's also a great one to ask everybody. So the question is, are you able to use a single lepidopteran for the PacBio 10x and HiFi, or do you use multiple samples? So I know both Marcella and Scott use multiple technologies to try and get these chromosome level assemblies. Are you splitting the insects to get that, or are you using multiple individuals?
No, the same, yes. For most of, yeah, for the four ones that I presented today, it's the same individual. For a few cases we have, but it's only if accidents happen, so mainly from the same.
Yeah, we've been able to go pretty low for the HiC step as well, using the ARIMA workflow combined with the Swift NGS prep kit. So, you know, something like the head of a fly, we've been able to get usable HiC data from that. And also when your assemblies are only in a few pieces, it gets to the point where the HiC data doesn't putting together thousands of contigs. You're just more validating the HiFi assembly and making some joins, maybe in centromeres or things like that, that the HiFi wasn't able to maybe assemble through. So it doesn't take necessarily as much or as perfect of HiC data to get relevant information to do the scaffolding at this point.
That's great to know. Next question is, could you imagine combining data from the low input protocol with data from the amplification-based ultra low protocol to improve assemblies of insects with maybe some larger genomes? Scott, I know you and I have talked about that previously and you were planning to test it. I don't know if you've tested it yet. We personally have tested that recently in-house and did get very promising results. So it is something we're looking into. So if you think about an insect that gives you maybe that 50 to 100 nanograms of DNA, and you're able to squeeze some low input and ultra low input data out of that, you might be able to, you know, use the longer reads from the ultra low and the coverage level from the ultra low in order to be able to assemble a larger genome. And that's what we see so far, but it's still in early days of testing it. I don't know, Scott, if you've played around with it yet.
Yeah, we've done some of the same sorts of things. I will say that, you know, if you don't have a lot of library or a lot of DNA to work with, it's very difficult to generate just the straight low input HiFi data. If you got 50 nanograms of DNA, you're not going to be able to get a lot of reads from that. But there are other applications where maybe you have, you know, 500 nanograms of DNA. You can generate a nice cell of HiFi data, but maybe it's just not going to be able to achieve the coverage. And so if it were me doing it today, I would hold aside 50 nanograms of that 500 for possible applications with amplification-based ultra low, and then come work together to put something in, especially if the genome size is north of 750 megabase or a gigabase in size. And so we're sort of getting into that now with some of these tiny things with multiple gigabase genome sizes.
Yeah, all right. And then a question that I would like to ask everybody is, what is the kit or technique that you're using for extracting the high molecular weight DNA from all of these organisms? Can you use bead mill to pulverize the insects before the lysing step? So maybe we'll start with Chris. How are you doing your DNA extractions?
So my organisms are tiny enough that I don't need to do any kind of disruption. They can be, less than 50 microns across sometimes. So I'm able to just pipette lysis buffer directly onto it, and it's not a commercial kit at all. It's just a salting out procedure, a very old-fashioned method using tris, salts, EDTA, SDS, Pro-K, and that seems to give us quite good yield of what's in there.
And Scott, what kind of methods for DNA extraction are you using?
Yeah, so all of our samples, usually they're samples that are shipped to us. They would have been snap frozen prior to shipment and then shipped on dry ice. We usually maintain them frozen and do pulverize them to a powder with a bead mill, making sure that powder stays frozen, and then add lysis buffer. We're favoring the Kaigen MAGATRACT high molecular weight kit, I think is what it's called, for doing these extractions. And our goal isn't necessarily to get 200 kb DNA, but just get something where you have very little fragmentation below 40 kb and a good yield. I think like what Chris is saying, when you're working with something tiny, you don't want to give up half of it to your extraction procedure. And so the Kaigen kit seems to do a pretty good job of doing that. With some oddball samples, we sometimes get the DNA still looking a bit dirty coming out of the kit, and then might do some subsequent cleanups with beads or some other approaches we have, so that when we go into shearing, the DNA is really primed to working well.
I will add to that, one thing that's been really important in getting high efficiency extractions is to use a carrier. And that's true not only when you're doing a pellets based thing like my salting out protocol, but also for sort of PEG, spry bead based cleanup methods. And I think it probably would even be worth trying to use carriers for very small insects to improve the yield.
Can I ask a follow-up question to that? What carrier would you use for your bead cleanups?
So I like yeast tRNA, which does have one downside, which is that although it's not as fluorescent to a DNA stain as DNA is, it does have some cross fluorescence, so it can interfere with quantification, but it's RNA, so you can treat it with an RNase and get rid of that after you've done your extraction. And that's not true of other carriers like glycogen.
Okay. So a couple questions about some specific examples. So for the Indian meal moth, they're wondering how much DNA you were able to get out of a single individual.
So that was one of our early trials with this, and so we probably had a microgram of DNA or so going into the low input. It's still, we maybe could have squeezed it on some sort of size selection, but went forward with it using the standard low input approach. And I think too, if you can shear the DNA well, kind of like what I was showing with little shouldering, one advantage of not size selecting is, in addition to, as long as you don't carry along a bunch of short reads, you're able to kind of bring along some longer reads where the ELF size selection or something like that just gives you that window, like where a very large proportion of your reads are between, let's say 12 and 15 kb, where when you don't size select it, you're able to carry in some of these really long reads into the sequencing, and I think they help sometimes in the assembly to kind of eclipse some repeat regions and things like that. So I almost favor not size selecting if you can, and if you can do it well, I guess. Yeah.
Perfect. And a very similar question for Chris. How much DNA were you able to get for the Castrella sample?
You know what? I don't even quantify it before amplification. I've tried this many times and it's pointless. With myofauna, even a femtopulse isn't sensitive enough unless you elute in something ridiculous like 5 microliters, and then you're giving up 40% of your sample for the quantification. It's nonsensical. What is really important is to quantify using that proxy PCR, and given some reasonable expectations about the genome size, you can tell pretty clearly from that if it's going to be a sample with, good library diversity and that's going to give you a good assembly. With things that are kind of sub 200 megabases, I would only be reluctant to sequence if I have to use more than 20 cycles. 16 is kind of typical for a small genome and something Castrella sized. But yeah, it's not necessary to quantify before amplification, and sometimes it's just spinning your own wheels.
Okay, perfect. All right, well, we're already past 11:15 here, so I'm going to go ahead and wrap things up here. I know there's still several questions, but I promise we will follow up with you after the fact via email. So I just want to thank everybody who attended today, and especially a big thank you to all of the speakers for those absolutely wonderful talks. As a reminder, this webinar was recorded, so it will be sent to you after the fact if you want to go back and listen to it. So check for that email coming from us in the next few days. I also wanted to let you know that if this has inspired you to do your own HiFi sequencing project, we are actually accepting applications for our 2020 Plant and Animal Sciences Smart Grant program right now. So co-sponsored by the BYU Department of Biology Sequencing Center. So please consider submitting your application at pacb.com/smartgrant, and we'll be taking those until July 24th. So with that, I'll say thank you, and everybody have an absolutely wonderful rest of your day or night.
Thank you.